um, doing the splicing can be really difficult. So I'm going to do an additional kind of run through, also kind of give you an idea of how I want the answers formatted. So this is going to focus on splicing, but the things I show you here might be useful in other parts of the project and in future projects. So I'm here in Eugene, and we're going to do a dot plot. So tools, build a dot plot. And if you remember, we found out in one of the previous videos that variant 2, all right, is what? Variant 2 is the one that doesn't have splicing. It's an RNA, all right, so we don't have to use that long stretch. We can just focus in on the RNA we want. So we're lucky that this exists. Doesn't always exist. So variant 2 had the whole thing. So we're going to look at variant 2, and we're going to compare that with, and this time I think I'll do variant 4. You can still do variant 4 if you want. This might make it a little easier. Or you could go back and use 5 or 3. And I'm going to do the thing. I am going to reduce this down here though to 50. Your exons might not be that long, so I'm just going to make sure that we at least make it shorter than in 1x. I'm not going to look at inverted repeats. No need here. I'm going to leave it at 100%. I know these two RNAs are the same. They should be. All right, so at least the sequences that came from the same genomic DNA. Hopefully nobody used a different organism or a different human to do this. Don't always know that, but I'm going to leave it at 100%, and we're going to see what we get. And yes, we get what we want. Version 2 and version 4 I'm doing here. So one is the longer one of 2 because it doesn't have the exon, so that would be this way. And version 4, or variant 4, sorry. Variant 4 would be this one, and it's missing this piece right here, so that would be right in here on this sequence. So it's kind of like this is version or variant 2 going this way, and this is variant 4 going this way. All right, so great. What can we do with this? It's nice and clean, at least, when we do it this way. All right, I can click on this one, and I can get this top bar up here, and I can grab exactly this. All right, and I can copy the selected sequence. All right, and I'm going to do what I told you never to do. I'm going to use a non-text editor. I'm using a more advanced one like Word or whatever. And there's a reason I'm doing it, but I'm going to show you where you're going to run into problems if you do it this way as well. So I'm going to paste it in here, and oh wait, I didn't get anything. Why am I not getting anything? Well, you have to paste unformatted, and now you get your letters. All right, and again, you won't have this problem in a less advanced text editor, but you might in Word or say something like this one. All right, well, I'm going to do that as well here. I'm going to click on that one, and I'm going to go up here to this top bar, and I'm going to right click, and I'm going to copy selected sequence. I'm going to come back here, and I'm going to put that under exon 2, unformatted. All right, well, great, but I'm really suspicious. Is this really the sequence I want? Well, just in case, you probably ought to go back and check it and make sure that it's the same as version 4 of your RNA. Okay, this is version 4. So, or both of them actually. It should start with 4 A's and it should end with a AGAA. Let's make sure that that is what we've got here. And so let's go look at our, let's see, this would be under downloads for me, wherever you saved the original. For here's version 4, AGAA, good, and it starts with A. Okay, this leads me to believe that I did this correctly. All right, so this one for version 4, it's probably the correct one, but you know, you might just go ahead and grab this thing right like this and copy it and put it in here somewhere, maybe above these, and then just literally print it out or use it on the screen. I'd print it out if you could. You know, make sure unformatted. Ooh, that's ugly. You can fix this by doing something like this, and you can change the font down to about 9. See if that didn't fix it. Yeah, that fixed it. All right, so we can go and we can compare these and make sure they're exactly the same. I would suggest doing that, um, but you may not want to take the time. I understand, but just in case, you might just kind of look through and kind of check out where everything is at, make sure it seems at the same spot. Do some different things. You can or can't do that. If you trust it, great. Now the other way thing to make for sure is, all right, what we're actually looking for are some pieces to annotate. So what could I look for? Well, one, I don't want to do it with, well, we can leave this up here. We know this one is correct. So why don't we do it this way? I'm going to go to view and I'm going to go to edit and find. Now your text editor may be different, but anyways, what are we looking for? We're looking for an AUG. So if we're looking for an AUG, here we're going to have to use T's. So it's ATG. Now we're in RNA, so this is fine. You don't have to translate it or anything or reverse translate it. So ATG. So let's find all the ATGs. All right, did it find any? Well, it did down here, which is great. What about up here? Where's our ATG up here? All right. Did it not start there? What's going on? Well, it only starts where your cursor was. My cursor may not have been here, so let's go see if I start my cursor here, if I find the ATG after I move the cursor back up. All right, so find ATG. Now it finds it. So make sure you know where your cursor is at. All right, but look, it didn't find this one now. It doesn't find all of them. This particular word processor doesn't find them everywhere. It just finds the first one. So what can I do to do that? Well, hit it again and it'll find the next one. That's the way this one works. You have to know the software you have. There might be a find all. All right, this one doesn't have a find all. It just find the next. All right, so we have to know something about the biology. Which one do we use, of course? Well, you use the first, in an ORA, you're going to use the first one. In other words, this ATG is the ATG I'm looking for. It's always, in an mRNA, the first ATG that comes along, that's going to be your start codon. So, pretty sure this is my start codon. All right, and so what are we going to do with that? I think this is my start codon here. So, I want to make this pretty so I can see it. Start is like a green light or a red light, green means go. I'm going to say that's the start, so I'm going to change the color of that to green. All right, and so now I have green ATG start codon telling me where it is. And I'm pretty confident that that's where it is. What you might be less confident in is where is the stop codon. Now the stop codon has to be in the reading frame, and that is very difficult to determine. And that's where we might want to go back to the to the Eugene, all right, and see what it says. But first, let's do it just like we did this way, just to see what we will get as far as the stop codon. So I'm going to change this to red because I'm going to change it to pink because we're not quite sure yet, and then when we're sure, we'll make it red. So the pink will be not quite red. All right, we're not quite sure that's the stop codon, red meaning stop. So for one thing, problem with stop codons are, oops, I didn't mean to do that. All right, one thing with stop codons are that there are 3 different ones. So we're going to have to search for all 3. Now, one thing to worry about is, well, one, we know it's in the second half. We know it's not up here. So we saw that if I click here, I'm only going to search after that. So what if I only search here? I'm only going to get the stop codons that are down here in this piece, and that's where I want them. So I'm going to do that, and that can save you some time. Might be dangerous, but you can certainly do it. And what's our first stop codon? Our first stop codon, let's try UAA, which would be TAA here. And we find one right there. Hey, that's not a bad looking one. So I'm going to put pink on there. All right. And then I'm going to find the next one, which is down here. And so I'm going to, again, make that pink. And then I'm going to find another. Oh, I found another one. So they're next to each other. This is good. Not all of your text editors find them when they're split up across a line. This one luckily does, and we can do it. But that could be one of your problems. You might have to go and make this all one straight line in one line for your text editor to work. This one, fortunately for us, or in this exercise, does. All right, and then we're back up to here. And we don't care about having one that's the same one. We're in the whole piece. I'm not annotating that. But it's good that it goes back up here and finds this TAA in between C and an A. All right, so let's go T and A. Yes, all right, found the same one. All right, great. So I found two potential TAA stop codons. We don't know which one is correct. The problem is we've also got some others to worry about. We also have TAG. So, okay, let's click here. Let's go edit. Let's go find. And let's go TAG. Nah, that's not one. That's too far up there. I don't believe that would be a stop codon. We've got others. I don't believe it. I'm going to go on. I'm going to assume that it's going to be later. It could be. There is nothing keeping that one from potentially being the stop codon, but I don't believe it. Well, because I think now this one could be. It's late enough. Who knows? It's past halfway. All right. And find the next, and then we're back up there. So, okay, we've got three potential stop codons now. Let's go and see how we might do the last one. So, again, I'm going to click down here. Now, we had one we skipped. Who knows? It could be. If it's in the reading frame, that might have been it. And I may make a mistake. But we're going to try and make some assumptions, and maybe that gets you in trouble, but I think I'm doing the right thing. TGA. All right, and I'm going to find. Still too early. I don't think that's it. Though, you know, if it is in the reading frame, as in 3333, it could be. But I'm going to go ahead. I'm going to go, no, let's wait. At least we get to the second half. That last half wouldn't have hung on as a part of this mRNA, you think, if it isn't. And we're getting closer. Do I decide I'm going to go one more? No. I'll go ahead and try that one. Ooh, there's another one. All right, that one. Ooh, another one. All right, and we're back up there. All right, so we now have several potential stop codons. Actually, this one did not. Hold on, this one didn't get chosen. But two in a row there after each other pretty much indicates if this one were chosen, that one would be chosen. So that actually doesn't matter. But all of those are potential stop codons. Our issue now is, are they in the reading frame? In other words, am I reading this one as CCT and then AGG? If it is, that's not a stop codon. If it's CTA and then GGG, that it's not a stop codon. But if it's read, so it's GCC and then TAG and then GGC, okay, that's a stop codon. So how do we do that? We got to do the reading frame. And the most straightforward way is to, of course, count them by threes. This is the stop, so it's this one and then this one, and then no, I'm not going to do that. Though you could. You know, one way to do this is not, or let's see, how could you do that? You click after this one and you put a space, and then space. All right, you could keep doing 3, 3. This is just the way this word processor works. I could do this. So I'm clicking here, space. Clicking here, space. Clicking here, space. 3, 3, 3, 3. You could keep doing that, and that would ensure that you're in the stop, you're in the reading frame. I really don't want to do that. Now realize splicing doesn't care about the reading frame, so you have to do it for the entire thing. All right, so I'm going to undo all of those because I really don't want to do, let's see, that's Control Z. I'm going to do Control Z and get rid of all that. There we go. And hopefully I didn't alter the sequence in any way. I can go back and check my ATG up here. So my ATG, where is my ATG up here? ATG, ATG, TGT. Okay, good. All right, so what's another way? Well, we go back to Eugene. Eugene is what we're looking for. The stop codon codes for methionine, all right, but if we go back and we look at our amino acid sequence, we're looking for a stop codon. Eugene is actually going to show us where the stop codons are. Now, to do that, we have to click on, let's see, where is it? Right here? No, which one is it? T, translate. All right, show all frames. Drop down here, and then here we are. And the stars are the stop codons. So we need to be in this second part. All right, and we see that it's the same. And what we're doing is we're looking for stop codons at the end. I'm down here. I need to be up here. So go to the end. All right, and we need stop codons. Now stop codons are these little stars. All right, we need the first stop codon in this piece. And let's see, how do we do this? I'm trying to get this label right here. Get it all, we've got, okay. So actually, it shows you it's a TAG, but that's starting there. Up here, all right, so it's, how do we know? How do we know which one of these is the actual stop codon? Well, we have to know the reading frame. And we don't know the reading frame down here because we don't know the reading frame up here. And to tell you the truth, the ultimate way to do this is to actually do like I did and put those spaces in, and that's the safe way. And if you want to do that, fine, that works. You do that and show me you've done that, and I trust whatever you've done. The thing we need to look at is not the longer sequence. We need to look at the shorter sequence to keep the reading frame in line. We know where the start codon is now. What we really need to do is actually go through that and do this exercise right here. So how can we do that? Well, I'm going to do a different dot plot. And what I'm going to do is I'm going to take that version 2. Actually, I'm going to take the version 4. All right, that's the one I'm working on. I'm going to do this. I want to open it, and I'm going to compare it to itself. All right, and I'm going to go 50, just like we did before. It really doesn't matter with one that's already there. All right, and I know where the start codon is. The start codon is, all right, we're looking at this whole thing. It's the first methionine in the sequence. So we are down here at 1. And so where is the first methionine that you run into? And it's here. Is this the one that we saw, that we chose? Make for sure. ATG right here. And before it is a C, and after it is a T. Let's go see if that's it. It's surrounded by a C and a T. Let's go look at our thing and see if that is indeed what we found. A C and a T. Yes. Looks about the right distance. That is our start codon. So that is our reading frame right there. All right, so now let's go back. We're looking at version 4. All right, we're in this reading frame. All right, now this one has already been spliced for us, so we really don't have to worry about it. We just have to go along this top line until we run into what? Until we run into a stop, one of these stars. So we're just going to slide along here. Let's see, I'm going to use, how can I do this? I'm going to use the arrows. Nope, I got to use the scroll. I'm going to try and, I do not, this is the one part I don't like about Eugene. All right, I'm at 1. Here's my methionine. All right, I'm going to switch over to the non-wrap. Is that it? Nope. I want the one that scrolls along the bottom. Let's see if I can get that. Aha, it's the top button. Sorry. All right, so here's our methionine. We're going to scroll until we see a star. Wow, we see a star at 220. Well, that would be the end if that's the case. Here's our protein. It would just be this little short one in between here. And it could be. If you make that justification, I'm good, because you know what? That is legitimate as far as we know. You know, and I don't see any other stop codons until way down here at 585. But why would we have that second piece going on here? Where is the splice site? The splice site, when we went and looked at that other piece, how many base pair were it? We should have looked to see, all right, where that splice site was. Is it after 220? All right, this little star right here, is it after that? So let's go back. Let's go back. Where was our splice site? So I'm going to redo our analysis. And I'm going to go to version 2, versus version 4. All right. I'm not going to do any of that. All right, no repeats. I will do this at 50. Doesn't really matter here. We're comparing it to itself. But I will do that. It's at 300. So you can make the analysis that, hey, this stops. This makes a tiny little protein. All right? And it actually doesn't use this piece down here because there's a stop codon there. Let's go see what that stop codon is, though. All right, so let's go back. All right, redo our analysis. I'm just looking at it against itself with version 4, just to check ourselves. There's no real reason to do that. But, all right, so we're here. All right, I'm going to click on the top one to get that one, and then I'm going to click that to get our RNA, our proteins. So we're here. We're in the reading frame that's on the top, and we're going to, I didn't make it. Do what I want here. There we go. I'm still in this. I hate this. I never can remember which button to push. Nope, show all frames. I'm good with just one sequence there. There it is. It's the top button. I always think it's the bottom button. All right. So here's our methionine, and we go all the way to 220, and here is our one that they give us the star. And it is a TGA. TGA is a stop codon. And there you go. Here's our answer. You tell me that, and you explain that this is odd because 220 is before the splice site, but according to all the rules we know, all right, this should be the protein, and it's a tiny one. But this would mean that it works that way with all of them. All right, all the variants should have that same sequence, but if you splice the way we do, you're going to get your protein at this with this size and this thing. And I will tell you that this is probably not the splice. This is probably not what happens. And there is probably, in other words, all I can tell you is sometimes the genetic code is not used correctly. And there are, depending on what's around it, you can't go by it. And the only way for us in this course to tell if indeed this was the case, all right, other than it sure does seem suspicious that you have a splice site after this, why would that be the case? That would mean go in the wet lab and investigate. Could be missequenced. There are lots of things that could have gone wrong here. But according to your analysis, if you want the grade and you just say, hey, that's the splice site, looks suspicious, but you know what? That's what it says. You get full credit here. However, let's go look, and we can cheat. We can go find the protein. So I'm going to do that real quick. We're going to see the protein for variant 4, and we're going to check to see if indeed it is that very small little protein that's made. All right, and we're going to check and see if the sequence looks the same as the one we just chose there. All right, so let's go to NCBI, nucleotide. We could go to protein, but I want to go to a specific one. And we want, we could go that way. Let's do it this way. Click here. Do what we've done many times before, GPX1. I'm just going to pipe in human. It should know. All right. But we can go to RefSeq proteins. Why don't we do that? Is there one that's tiny? Look at this. Isoform 2, 98 amino acids. Isoform 1, isoform 5, we were looking for 4, 170 amino acids. So I can go to this one and look at the nucleotide. Let's go look at the nucleotide version of this one. All right. Oh, it gives us, I don't want to do that. Gives us the thing. Let's look at the FASTA. It's pretty tiny. Maybe this is what happens. All right. So let's look at these last couple of amino acids. CCLEE, LEL. All right. Is that what we get here? Now, this is the way you can cheat. The answers are out there. I'm telling you, you can find the right one. So let's go back to Eugene. Let's go to this this one. No, this is not the sequence that we get. All right, but now that we know this one, you can actually go find the correct answer. But if you tell me, you have to logically tell me why you chose some of these things. And telling me, well, that's what it said, that usually doesn't cut it. But you can cheat and find the right answer here. So where is this? Let me go back to, what was it? I want where is CCLEL in our sequence? All right, in isoform 4. So that's what we want. I got to remember that. CCLEL. CCLEL. Well, if we scroll down, we're looking for a CCLEL somewhere up here. Now, I think it said, what, 170? So multiply that by 3. That's 170 times 3 is about 400 and what? 510. So somewhere around 510 along here. So it certainly doesn't go near the end. 510. CCLEE. I don't see it along here. Alright, that doesn't make sense. So let's go back. Let's look at our thing. It's a... How many amino acids did it say? It was on the previous page. 170 amino acid protein. And we're looking for, you know, LEL, CLEL, something like that. In Eugene. And then a stop codon. So little stars are a good place to stop. So you can use these stars and look beforehand to see where it is. So let's go all the way to the end and we'll work backwards. But I know it's not this long because that amino acid sequence was only a few hundred. So we should be able to do it somewhere around, I don't know, 500. Ah, there it is, right here at 585. Well, what we forgot was that there was some sequence before here, so the amino acids start here, so you had to add on 75 in front, all right, and then you go through, and what we're doing is we're looking for that end, and you can find it. Let's see, where was it? Well, you know, it was 500 and something. 545. 590, where was it? See, it was a star, right there it is. And it is in the correct reading frame. So you can start here, but we go backwards, and you're going to notice that even though that's in the correct reading frame at the top, no, that is correct. There's a methionine at 250. And there's a methionine here at 75. But we get a stop codon here for some reason that doesn't get used at 220. And there's why you have to have wet lab experiments. So in other words, am I expecting you to get the exact right answer? I've shown you how to cheat now and get the actual known answer from the wet labs. But you can see that through our analysis, this is incorrect. You're going like, well, what good is bioinformatics? Believe me, it's useful. You can basically cut down on your errors and use it to explain your wet lab experiments. But sometimes it doesn't fit with the theory. All right? And so don't get frustrated. If you give me a rational answer, all right? So here, if we were doing this the rational way, and you said, well, according to the theory that we understand, all right, we start with methionine, all right, on this, and then the first one in the reading frame, before we get to any splicing, we got to a stop codon, then that's the protein. That is a logical answer. That is an A+ answer. How can you know that just for some strange biological reason, it decides to not stop right here? And it doesn't, all right? It continues going, and it goes on to the one over here that is on the other side of the splice site. So hopefully this helps you a little bit. I mean, I know it's confusing. I didn't say it was going to be easy here, but if you know the rules of how transcription and translation work, it should tell you what you should get given these sequences. You may get it wrong, you may get it right, um, you can go check yourself with the real answer, which we just did, which makes no sense. Why did it ignore that stop codon? But it did. All right. And there could be reasons for that. For one thing, there's this CECUS thing, all right, in this protein. Maybe that affects it. That isn't what affects this one, but that it could be a possibility. It takes the stop codon and replaces it and keeps going. So keep that in mind, that you never know what the real answer is going to be until you get into the wet lab. But if you do this analysis and you give me logical reasons for why you chose the piece that you chose and said that that's the stop codon. So according to me, if I'm doing this and I'm turning this in, I'm going to analyze this, and I'm looking at this up here, and this here, the first stop codon is before the splice site, so we didn't change reading frames. I didn't have to change from here to here to here when I spliced, because you can splice in the middle of a codon. There's no reason you can't. Then I would just say that that's the stop codon right there, and it would be a TGA, and it's going to be actually in the top one. And this whole idea of me choosing the ones down here was wrong. I said, hey, I thought these were good candidates, but they're not. There's one up here that is a, what was it, a TGA? All right, let me go back. Where was it, Eugene? TGA. So let's see if we can find that one. TGA, let's see what's on either side of it. TGA with a C and a G on either side. So I'm going to go back up here. Let's see. And I will start there, find, edit. TGA. But we can cheat a little. I'm going to go back and I'm going to, what was on either side? A C and a G. All right, so I'm going to go back here. I'm going to put a C and a G on either side. And there it is. All right, so there's your stop codon according to the standard rules that we know. What can you do? It's in the reading frame that was started by the ATG, but they're splicing on the other end of it. And obviously an amino acid goes there, and it is not used as a stop codon. Sometimes we don't understand why this occurs. There are people who study this, but you're going to see this with the lac-operon as well, where the rules are just not followed. So just giving you, only makes this harder, but I grade it in such a way as you give me a logical reason for this to occur. And a good logical reason is that, hey, this is an odd gene. It uses this thing called the CECUS, and it replaces stop codons with a new amino acid. Maybe that's what happened here. And you can make that good argument. You need to read about CECUS to answer that, though. Now we go down here, and I want to give you another hint. That often stop codons will come in pairs, and that's usually the one that is used. So there is a good argument, and I just happen to know that. You don't know that. But I just happen to know that stop codons often come two in a row, and that's usually an indicator that that's the correct one. And there we go. We have two of them in a row here, so that is very likely that could be a stop codon. And after all, why not put it at the very end? And this is probably a stop codon used in one of the other variants. In other words, in one of the variants, I suspect that this is absolutely the stop codon. It also might have to have something to do with the CECUS element. You need to read about that as well. All right, if you're going to do the extra credit, you do. So I'm just giving you some hints for the extra credit. All right, so keep that in mind. Now, how do you find the intron again? Well, go search for this, and go search for this, and pull out the pieces in between and place it in the intron. Now, there are rules for the intron. Once you find this, you find the closest thing you can to the rules. So you read the rules, and once you have the intron placed in here, all right, then you just look at where it says you should look. You know where the splice site is. You've got that. And you look in this area, for the splicing signals. All right, you look for anything, the closest thing you can find. If you can't find it, that's legitimately. Sometimes you just can't find these things. This is such a fuzzy art, and it's amazing that people just let the computer run through it with the rules and they expect to find the right answer. And people publish things, and it is just amazing. Now, this is useful because without doing what we're doing right here, you are going to do, more than twice, 10 times the amount of lab work to actually figure out what's going on. If you don't do the bioinformatics ahead of time, you have taken what might have taken you 6 months to figure out in the lab. You're going to take 2 years to do it. And, it's amazing that you have to use the bioinformatics, even as imprecise as it is, with the wet lab in order to find out what these sequences do. And this is a true art, as you can see. I've done it, with sequences, and I do it for classes here, and I take knowing what the answers are, like we did here, and then working backwards. How would you get to that without going into the wet lab? And sometimes you can't. All right, just plain and simple. There is no way, and it's practice, and you have to use it in conjunction with your wet lab. All right, that's it for this one. I will be happy, more than happy, and I'm sure this is really confusing to you. It is every semester. I will go and help anybody on Zoom. Usually I can do this in person, but I have no problem helping any of you on Zoom, and we'll get it so that we can share screens. I'll log in correctly so that we can share the screen and I can show you how to do this personally. But please, don't get too frustrated. It is hard. All right. Hopefully this one helped you more than the others. But you can also see how I want you to format this. I like this. You can color it outside of the regular text. Just remember all the kind of pitfalls we ran into here with doing this. And then, once you have them color coded, you can come down here and say, when you turn this in, green equals, do something like green equals start codon. All right. And, highlight it green or something. All right. And that's good. That shows me green is the start codon. You put it there. And so you've annotated this. And this is the form I want. Now, for this one, you would go, okay, start codon. When you go to explain it, you explain why you chose that start codon. So, why did you do this? Well, what were some of the things? Well, what are the rules? This was the first ATG from the 5' end. So the 5' end of the mRNA is, this was the first, ATG moving, how would you say that? Moving 5' to 3'. And that's the rule. The rule is the start codon is the first ATG you run into, and this sets the reading frame. All right, until the end of the splice site. All right, so sets reading frame. Until splicing. All right, that would be something you might want to tell me. And that'll give you a little bit more. I'll give you more on how to format it, but this would be the one that I think will help you the most. All right, this is kind of the. I was going to do this later until you kind of worked on it some, but if you can do this is start and stop codon. You can also, you'll have to tell me why you think the splicing is there. Go through the splicing rules, and look here. Oh, is there something here that's interesting? Is there something here? And then, of course, inside the intron, there are pieces you need to look for. And you find the ones that you think are the most likely. How do you know where these splice sites are? You don't unless you go into the wet lab. All right, and you extract the RNA. You there are the RNAs that are produced, or you interrupt you extract the nucleus and literally pull out the RNAs that are in there, and then look for the RNAs that are present, and you find out which ones are actually there, and you find these variants in there. Really tedious lab work, really tedious, but you know what to look for if you've done a little bioinformatics, and that'll speed you up on your RNAs. You'll be able to tell out, okay, here's the one that does this. I've got 4 variants. I've got, okay. And so this is not easy work. And it's frustrating because biology doesn't always follow the rules. There are the trends. Maybe they shouldn't be called rules. They're trends. One of the things we're going to find in the Lac-Operon is that ATG isn't the only thing that can be a start codon. And that's really frustrating. And but that happens in bacteria some. Not as much in eukaryotes. In bacteria, that happens. All right. So hopefully this helped. And remember, I will help you as much as I can. We can Zoom.